Well, our last speaker of this absolutely terrific symposium on viral etiologies of cancer is Arnie Rabson, who is from UMDNJ, the Robert Wood Johnson School of Medicine, and the Child Health Institute there. He's going to tell us about HTLV1. He's going to talk about pathogenesis, infection, and its role in cancers. Thank you.
Thanks, Arnie, and thanks again for inviting me, and thanks to all the organizers of the meeting, to Ned and Suzanne Kristen and the folks in Sunita and Nancy and the Cancer Institute office. It's really a pleasure to be here, and I appreciate all of you coming back from the tea and from the cookies for this last talk, so thank you. We're going to talk about the last one on that list of viruses, the only retrovirus so far on the list, even though retroviruses in the animal kingdom are very large representatives of naturally occurring oncogenic viruses. There really is only one oncogenic human retrovirus at this point, and that's the human T-cell leukemia virus, type 1. I'm going to start with acknowledgments in case I run late. I don't want to forget the most important part. Most of the work that I'll talk to you about today was done in collaboration with, oops, I just did the same thing. I have to teach myself this. Was done in collaboration with Yufeng Shi's laboratory up at Robert Wood Johnson Medical School, a wonderful immunologist. Alison Swames-Tusky did a lot of the work that I'll tell you in collaboration with Yufeng. Xin-Ching Lin really founded most of this project, and I'll talk to you also about work that Céline Granier has done recently on some of the tumor models. Oh no, there we go. Okay, I have to learn this too. You're not alone. Okay, so we'll start, as other speakers have done, with the tumor itself. And this is an interesting bit of history. HTLV1 was first identified really as being associated with a tumor that was first found in the late 1970s in Japan. And again, sort of like the EBV story, a T-cell lymphoma was noted in the southernmost regions of Japan by a number of investigators, particularly Uchiyama and Hinuma, who noticed this clustering of very aggressive CD4 positive T-cell lymphomas. And markers for T-cell surface antigens were just being described. And in fact, several laboratories were identifying the Waldman lab, for example. Uchiyama was involved in that, identifying the IL-2 receptor alpha chain. And these were activated T-cells, CD4 positive, with the IL-2 receptor alpha. And this clustering immediately did suggest, again, the potential for an infectious etiology. Cell lines were developed, and sort of like the stories we've been hearing in other systems, those cell lines reacted with sera from patients, suggesting a possible infectious etiology that the patients were developing antibodies to. Again, high levels of IL-2 receptor. So this sort of work characterizing the disease was going on at the end of the 70s, and in the early 1980s, about 1980, in parallel with this, based on his discovery, one of several people really working with discovering the T-cell growth factor, Gallo, Poise, and Russetti, were looking at CD4 positive T-cell lymphomas that were called Sézary syndrome or cutaneous T-cell lymphomas, and identified one that had budding retrovirus by EM and reverse transcriptase activity, the first human retrovirus identified, which they dubbed HTLV1 for human T-cell leukemia virus 1. And very quickly it was clear that HTLV1 was the causative agent of the clustered aggressive CD4 cell tumors in Kyushu, Japan, and elsewhere in the world. The tumor itself is a very deadly malignancy. The full-blown acute adult T-cell leukemia lymphoma, untreated, has a median survival of somewhere on the order of 3 to 4 months. Patients can go into complete remission with combination chemotherapy, now with bone marrow transplant, also importantly with antibodies, cytotoxic antibodies, but particularly with the chemotherapy approaches, rapid relapse occurs, and we may talk about that a little bit more later because there's some interesting features to the relapse. The disease often presents both with plaque-like skin lesions, which is where there's a confusion with the related Sézary syndrome or cutaneous T-cell leukemia, cutaneous T-cell lymphoma, which is not actually HTLV1 associated, but there can be some diagnostic confusion. There clearly can be a leukemia, a lymphomatous phase with involvement of lymph nodes and solid organs. And the sort of pathognomonic feature of this disease are these flower-like lymphoid cells, these malignancies with multi-lobar nuclei. Others look more like T-cell blasts, but there's a range of these sort of cells, and particularly the flower cells as sort of the diagnostic tip-off for this disease. There are a number of clinical features that those of you who've interacted at all with patients are very aware of. Very high white blood cell counts, hypercalcemia, and then again involvement of lymphoid organs as well as the leukemia, and all kinds of different sort of subtypes, a more smoldering type or a more chronic type as well. But again, what is common in the evolution of this disease is an oligoclonal T-cell proliferation evolving to a monoclonal T-cell proliferation. We'll talk about that more later. Early on, it was clear that these patients have deregulation of multiple different cytokines. They're very highly active T cells spewing out large numbers of different kinds of cytokines. And in fact, patients can have associated immune suppression and opportunistic infections. And in fact, in a very famous set of papers in June of 1983, I believe, it was proposed that HTLV1 itself might have been the cause of AIDS at that point because of the immunosuppression. The ATL has clonal integration of the HTLV virus, again, seropositivity, and the cells themselves are markedly aneuploid. The chromosomes look like you took a blender to them, basically, and we'll talk more about that in a few minutes. HTLV1 itself is a delta retrovirus. These are complex retroviruses. We'll look at the genome in a few minutes, but multiple open reading frames. The diseases are specific for CD4 cells. There's some infection of CD8 and macrophages and monocytes as well, but predominantly a CD4 infection, and the diseases are CD4 based. About 95% of patients who are infected remain asymptomatic through their life. Okay, and there are an estimated 15 to 25 million infected people in the world. But again, fortunately, very few people develop disease. There are regions of endemicity in Japan, in the Caribbean notably, and now more and more in Brazil, also in Africa. And this family of viruses, the delta retroviruses, also includes a related HTLV2, human T-cell leukemia virus 2, not associated with any clear disease. Simian viruses related to both of them, and very interestingly, an epidemic bovine leukemia virus that causes leukemias in B cells in cows and is sort of endemic in parts of Europe, for example, and has a lot of similarities in how the disease develops to HTLV1 disease. So as I alluded to, most people infected with this virus get no disease. Of the people who get disease, there are different forms or presentations of disease associated with infection. We've talked about adult T-cell leukemia lymphoma, a lifetime risk of about 2, 3, 4, 5%, somewhere in that range. Varies a bit with the studies and the populations, but 2 to 5%, with a clinical latency, period between infection and development of disease, of on the order of 20 to 60 years, okay? Long latency between infection and disease development, consistent with a lot of the concepts, as you'll see, that we've talked about, Dr. Sugden talked about with EBV, with multiple other cellular changes involved in progression of disease. Very interestingly, in sort of the mid-1980s, groups from both Japan and France identified a strong association, in fact, causal association, of HTLV with other sets of disorders, particularly a neurological disorder called HTLV1-associated myelopathy or tropical spastic paraparesis, which we'll talk about in a moment, but an autoimmune-like disorder, as well as other autoimmune-like disorders such as uveitis, arthritis, some patients with Sjogren's syndrome. So a spectrum of different kinds of diseases associated with infection, and again, some patients with immunodeficiency. So HAM-TSP, the neurological disease, is a disease that comes on months to years after the initial infection, so a shorter time frame, usually in older patients with infection following blood transfusion or sexual transmission. So what I didn't point out before was ATL, the leukemia lymphoma, often occurs decades after an initial infection, which is most frequently postnatal particularly by breast milk. So breast milk transmission to the early infant associated then with a long clinical latency period and then ultimate development of the malignant disease. Ham-TSP and some of the more autoimmune-like diseases occurring more rapidly and generally associated with blood transfusion, sexual transmission. So this is a chronic, slowly progressive spasticity disorder, muscle weakness, that's the paraparesis, can progress to bowel and bladder dysfunction, but generally is not fatal. Generally is a progressive disease controlled by steroids. Patients, again, seropositive for virus with antibodies in cerebrospinal fluid, but also, interestingly, in cerebrospinal fluid is oligoclonal proliferation of CD4 cells, as well as CD8s. So there's CD4 cell, infected CD4 cell oligoclonal proliferation, as well as a T cell response to that. And you can see atypical lymphocytes again, and again a demyelinating disease. So both diseases characterized by oligoclonal T cell proliferation of infected cells. So this summarizes what I just said. HTLV1 infection of the neonates can either be asymptomatic throughout life or a long latency period and then development of leukemia lymphoma, or infection later in life with a short clinical latency period and development of disorders such as ham-TSP. So this is what really got us very interested in this virus, having studied HIV for years, where there's a clearly variable but fairly stereotypic and predictable response following viral infection in terms of what happens in the host. Here is a virus that does so many different things. It does nothing. It causes virulent malignancies, or it causes these funny autoimmune-like disorders. So the grounds for studying sort of the viral-host interactions in pathogenesis are extremely interesting and fertile, we believe. So in terms of the pathogenesis of ATL, we've talked about the idea of oligoclonal T cell proliferation preceding the onset of the monoclonal leukemia. And I don't have on the slide, but I want to parenthetically mention that patients who go into complete remission from their ATL, who then relapse, there are several documented episodes of them relapsing with a different clone. Okay, a different integration site, a different clone. So there is this sort of pool of oligoclonal cells waiting to expand when the right other events happen. And we talked about the effects of age, et cetera. And as I'll show you a little bit more later, HTLV1 itself will immortalize CD4 positive T cells quite efficiently. This is very much analogous to the EBV B cell story. And the immortalization actually proceeds from an IL-2 dependent to ultimately an IL-2 independent state with activation of multiple cellular signaling pathways. So this really just makes that point of an initial CD4 positive T cell infection, expression of the HTLV oncogene, we'll talk about in a few moments, with a polyclonal proliferation, oligoclonal proliferation, and ultimately the monoclonal proliferative event leading to tumors, and clearly development of multiple cellular mutations, translocations, chromosome loss, tremendous genetic instability associated with the development of disease. So we've been interested in for a while in what are the possible determinants of these different effects of infection. Could the determinants be viral? Is it a viral strain, one strain that's neurotropic, for example? There is no evidence really at this point that supports that the virus strain or virus sequences really play a role in the different kinds of disease processes. So that really appears to be not an issue. Obviously, host factors then become the issue, and the issue of the interaction of the host and virus. Now, genetic factors are just beginning to be examined in different ways in terms of genome-wide associations, et cetera. So far, the only real association has been some association of MHC types with ability to infect the infant by breast milk ingestion. So certain MHC types in the infant are associated with more likely to actually become infected, but very little in the way of clear data. The host immune response, though, clearly plays a role, as we'll touch on as we go along. And we, for years, dating back to our HIV work, have been interested in the roles of virological and clinical latency and how those interact in pathogenesis, both in HIV and now in HTLV1. So very briefly, retroviral life cycle, most of you are aware. HTLV binds to the surface of the target CD4 cells. The receptorology of this virus has been very complicated, and in fact, many postdoctoral years have been burned in less than fully successful efforts to find receptors. There does appear to be a complex now involving a number of different proteins that appear in activated T cells. So virus binds, is taken up, reverse transcribed, and integrates. Following integration, we can have the classic sort of productive infection with new virus produced, and this virus does spread almost exclusively by cell-to-cell transmission. Fortunately, unlike HIV, this is really a poorly infectious virus. You don't have free virus floating around sort of ready to leap in to other cells. It seems to be a somewhat unstable virus, and there are also determinants after uptake of the virus that are fairly weak in terms of some of the early steps. So it's not a highly infectious virus. Cell-to-cell transmission is really the most efficient way to get infection. But clearly, as with all retroviruses and apparently essential, most, if not all, oncogenic viruses, the ability to enter into a latent state of infection with very low levels of viral gene expression is part of the life cycle of HTLV1 as well. The genome is a complex retrovirus with the promoter elements, the long terminal repeats at either end, classic retroviral genes, GAG, POL envelope, defining all retroviruses, and then a series of regulatory proteins, including one not shown that's actually from the opposite strand, going in the opposite direction, that are involved in different aspects of regulating the viral life cycle and the host response to virus. And we'll focus most on TAX, which is the transforming protein or oncogenic protein of HTLV1. TAX actually interacts with sequences in the viral promoter, the LTR itself. It's a transactivator that turns on viral gene expression through interaction with TAX response elements in the promoter, and there are multiple different TAX response elements. Again, a complex retrovirus, meaning that there are multiple different spliced isoforms that can give you full-length, envelope, or different variants of the small RNAs as well, including messages for TAX and for an RNA processing protein called REX, similar to REV and HIV, as well as these other regulatory proteins that have different roles in the viral life cycle and in host response again. TAX itself, with respect to the virus, interacts with transcription factors, specifically CREB-ATF dimers, forming a complex that recruits in co-activators, interacts with polymerase itself, evicts nucleosomes from the LTR, and turns on transcription of the virus itself through the LTR fairly potently, actually. And just as an aside, I meant it was on my acknowledgement slides. A lot of the details of this sort of work were worked out in some of the real details by a wonderful molecular viral oncologist named John Brady, who died yesterday. And John, some of you may know, was also instrumental in really understanding a lot of the SV40 initial regulation as well. So I just want to make sure to credit John and in a sense, dedicate this to a really wonderful man and a really great scientist. Peter, I know, worked next door to him for years as well. So HTLV TAX. So TAX, when I first started describing what TAX did to my former mentor, Mal Martin, who was an SV40 virologist for many years, Mal said, this is the T antigen of our time, meaning, in fact, that TAX is a 40 kilodalton protein, does an immense number of things, okay? TAX turns on, works through CREB-ATF proteins, as I told you in the cell, turning on the LTR, and also multiple cellular genes. It interacts with other leucine zipper factors, including serum response factor, which activates CFOS, so pro-proliferative. TAX, similar to what we heard earlier about KSHV, TAX interacts with the NEMO subunit of the IKK complex, the NF-kappa B activating kinases, and turns on NF-kappa B, turning on then multiple different cytokines, turning on a whole array of anti-apoptotic genes, as well as a subset of pro-apoptotic genes. So TAX, in fact, can be strongly pro-proliferative, interleukin-2 being very pro-proliferative and its receptor for T cells, CFOS, etc. So it can be pro-proliferative and anti-apoptotic, you know, your perfect recipe for oncogenic activity, but it also can be pro-apoptotic, and Céline Grenier is working on that in the lab as well. TAX can repress transcription of some cellular genes, including some of the genes involved in DNA repair, and in fact, TAX also inactivates P16, cell cycle inhibitor, CDK inhibitors, promoting cell cycle progression, and it inhibits the mitotic checkpoint, giving rise to these chromosomes that are a complete mess. So it really is a combination of activities guaranteed to transform, if you sort of reach a certain threshold or a certain hump with this. So now what about the latency issue that we've been involved in for years? There's sort of this tenuous, not scientific, but intuitive connection between the idea that there are long periods between infection and development of disease and the idea that virus can maintain in latent form in cells as well. And is there any connection between these, between the latency issue and the pathogenesis issue? Again, a common theme, for example, in EBV and others. So in vivo, it turns out it's very difficult to detect HTLV1 RNA or protein in both patients who are infected who don't have leukemia, as well as in leukemia patients, even though the load of viral DNA, the number of cells containing integrated viral DNA, can be very high. But if you take patient cells and put them into culture, you get rapidly, within 4 hours or so, abundant expression of HTLV1 gene expression. We and others have seen that if you take cells directly out of patients and grow them as lines, you can maintain them as latently infected cell lines, and I'll show you some data about that in a minute. But on the other hand, there are very high levels of cytotoxic T-cell response to this virus in vivo. So patients clearly are continuing to generate antigen, supporting an active, ongoing, continuing cytotoxic T-cell response. And just parenthetically, the CTL activity may be very important in the neurological diseases, where there may be this spewing out of all the cytokines and, that are the products of these activated CD8 cells. And that's certainly one of the major hypotheses in the neurologic disease. So this is work now that dates back many years that Xin-Ching Lin in the lab really sort of pursued, and it was looking at a series of different cell lines, and lane 1 in each of these are sort of the basal expression of TAX shown here in the cells, showing very low levels of TAX expression in multiple different cell lines, but different stimuli in different lines could induce TAX expression. So lane 4 are histone deacetylase inhibitors, not surprisingly, lots of TAX expression. Excuse me. But lanes 2 and 3 are PHA and PMA, so activation of different aspects of the T-cell receptor signaling pathway. So the hypothesis quickly became that T-cell activation of latently infected T cells might be sufficient to induce virus expression. Very similar to some of the points that came up in response to Lori Covey's question that Dr. Sugden talked about B-cell infection and the relationship to B-cell activation. So in this setting now, what Xin-Ching did was use antibodies to the T-cell receptor, classic mode of activating through T-cell receptor, showing again marked activation of TAX expression in these latently infected lines through T-cell receptor activation. This could be shown at the RNA level by experiments that Peter Simon did. Actually, in this case, he was using PMA working downstream. And what he showed was that this was a control gene that doesn't get turned on or activated by PMA. This is arginosuccinate lyase, which turns out to be a good control because it's not activated in T cells either by T cell receptor or by TAX. TAX activates probably over 3,000 cellular genes, so it can be somewhat problematic. Even actin promoter is turned on a bit by TAX, so it's problematic to find good controls. But when you treat with PMA, you get very rapid induction of a series of different mRNAs, and this is TAX RNA. Within minutes, you have high levels of TAX RNA. RNA for the full-length virus takes a little longer to come on, as does envelope, and this is really characteristic of the complex retroviruses, where there's an early activation of regulatory genes followed by a later induction of structural genes. This is sort of puts these complex retroviruses into the same kinds of regulatory schemes as the DNA tumor viruses, with early regulatory and late structural activity. And this, again, Peter showed at the protein level as well, very early induction of the short regulatory TAX and REX messages, with envelope coming up later as well, quantitated here. So this is Western blot for protein. So does this schema of latency activation through T-cell receptor activation, could it have an effect in thinking about how disease would work? And parenthetically, let me just make a point that I think also bears on some of the thoughts that we talked about with EBV and even with KSHV. We used to say in the HIV world, where HIV is activated by cytokines associated with inflammation, that what the inflammatory stimuli do is provide new target cells for when the virus comes out. So you link inflammation with exit from latency, just like a phage system just about, where you link production of virus with production of target cells through activation, inflammation, et cetera. So if the virus comes out, it's more likely to find a target cell than to be destroyed by proteases in the blood or in this extracellular space. So on one level, we would hypothesize that that's what's going on in the HTLV system. But on another level, can this concept of immune activation of virus expression, particularly of the TAX protein, can it inform our understanding of the pathogenesis itself? So the model that we're considering and that I'll explore with you in the next few minutes is that you have an initial infection of T cells in vivo with spread of virus to many, many T cells. That probably initial infection, probably millions of T cells initially infected. Strong cytotoxic T cell response, particularly to TAX itself, so a strong selective pressure for latency. Okay, it's similar probably to some of the EBV issues. Strong selective pressure for latency. So now you're floating along for years with latent infection, but say you're a T cell who now sees its cognate antigen or a superantigen, and you have immune activation now leading to induction of the TAX protein. TAX will lead to proliferation of activated cells. And at that point, either you can have a successful anti-HTLV CTL response and either control infection, remain asymptomatic, or potentially move into the autoimmune disorders, or with additional mutations begin down this track, leading ultimately to the malignant disease. So that's the model. So how do we study that model? Well, Peter Howley will remember this in great detail, I suspect that Arnie will as well. In the mid to late 1980s, George Corey and Gilbert Jay in his laboratory and a number of talented postdocs wanted to sort of take the new transgenic mouse model system that was just being developed and figure out how HTLV1 worked. There was a new focus on lymphoma in the lab at that point, away from SV40. So they made a transgenic mouse, or a set of them, that were a mini virus. It was the TAX gene being driven by its own promoter, so it should feed back and turn on its own expression, okay? And in the first set of founder animals, in fact, several of the founders died at 3 to 4 weeks of age, and they couldn't be propagated. And those founders had a lot of expression of TAX in their thymus, okay? And they probably had sort of a cytokine storm. They couldn't be propagated. There were 3 founders that survived that over time developed these very characteristic neurofibromas on their tail and on their ears. And they really look histologically like human neurofibromatosis, actually. They had no expression of TAX in their T cells, and they don't get T cell lymphomas. So our reasoning was that these mice are a mouse model of latent HTLV1 infection that could potentially help us look at the model that we have for pathogenesis. So what Alison Swaim-Tusky has been doing then is beginning to really examine these mice immunologically. And curiously enough, no one had actually, after that first paper which used a northern blot to say that there was no expression in the T cells, no one went back and revisited HTLV TAX in the T cells of these animals. Nobody had looked at it, surprisingly. So Alison went back with Yufeng Shi and first just started to characterize the T cells. And what she saw is when she put the littermate controls versus the TAX mice cells into culture with IL-2 and following anti-CD3, T cell receptor activation, the TAX cells were hyper-responsive. Within 24 hours, they're proliferating compared to control, they're clumping, they look like activated T cells, and even at 48 hours, they're far more proliferative than the control cells, which take to about 72 hours to begin to proliferate. This proliferation, hyperactivation, is associated with induction of expression of the TAX mRNA, similar then in the mouse cells to what we saw in the human, and induction of activation markers such as IL-2 receptor alpha and CD69. So the TAX cells are hyperproliferative, hyperactivated in response to T cell activation. Alison has a very immunologic bent, so she's interested in what kind of CD4 cells these are. And I think most of you are aware that there are multiple different forms of T helper cells, Th1 cells that make characteristic cytokines, interferon gamma, TNF, Th2, more involved in supporting antibody response, pro-inflammatory Th17, and the Treg, or repressor type T cells. So Alison embarked on a series of studies to look at what kinds of cytokines are induced. Control cells in this setting tend to bias towards that Th1 cytotoxic response. But interestingly, and perhaps predictably from some of the patient material, the tax cells make tons of Th1 cytokines very early and persistent. They make some Th2 cytokines, much more than control cells. They make Th17-associated cytokines, as will come up as well, so IL-6 and IL-6 and IL-17 at different time points. And they also make cytokines associated with some of the T regulatory functions, like IL-10, and also GM-CSF, an activating cytokine. So these are essentially sort of multi-potential cells exhibiting features of all of these different types of T cells. Now, here was the kicker in Allison's experiments that's really sent us on our way with the model. Normally, if you take control littermate cells and you stimulate with anti-CD3, anti-CD28 in culture, the cells proliferate like crazy for about 2 weeks or so, and anyone who's done these experiments knows that the culture undergoes a contraction, and essentially all the cells die within 3 to 4 weeks. It's a very stereotypic apoptotic response that happens. In now somewhere over 10 or 12 different mice that we've done this experiment, a single round of anti-CD3, anti-CD28 stimulation, and anti-CD3 alone is sufficient, leads to immortalization of these cells. Allison has kept the cells over 65 weeks in culture. So a single round of stimulation leads to immortalization. And that immortalization is accompanied, first of all, by persistent, though somewhat variable expression of tax. We've taken it out to 18 weeks, again, with sort of an up and down picture of tax expression, but continued tax expression, associated with CD25 expression, activation like patient cells, and associated with loss of CD3 expression, T cell receptor, which is also true in ATL cells, that ultimately the real tumor cells lose T cell receptor. Allison has begun to ask, or at the beginning of this, what are some of the molecular events that might promote this long-term survival? And she's looked by a qPCR sort of array analysis at some anti-apoptotic genes, and in particular, MCL1 expression is activated even at 72 hours after immune stimulation as compared with control cells. There are other genes as well that are turned on, and a number that are turned off. There's a bit of an artifact here that some of these are turned off more than 4.8-fold, but the quantitation cut off there. But MCL1 became a focus because it's such an important anti-apoptotic gene, and it's an important anti-apoptotic gene in T cell development in vivo as well. Surprisingly, the role and identity of MCL1 and HTLV1 pathogenesis really hasn't been known. So far, what Allison's been able to show is that at the level of RNA, MCL1 is activated early and persists particularly through this time point when the normal cells are dying, persistent expression out at a month or so, but in very long-term culture, she loses MCL1 expression, and here at the protein level, again, at 2 weeks, high levels of protein control compared to control. So we'll be following up on this, obviously, but MCL1 is a possible mediator of this long-term survival at this early stage of the tax cells. Now, are these cells in any way oncogenic? Allison did a very classic adoptive transfer model. So she took stimulated tax CD4 positive T cells, actually she's done it at late time points, but even as early as 2 weeks after the stimulation, and inoculated them into the highly immunodeficient NOG-SCID mice. And in those mice, what she gets are proliferation of these cells with a wasting disease that ultimately we have to sacrifice the mice at about 2 months, about 30% of peripheral blood cells, all leukocytes. 32% of leukocytes are CD4 positive. It's essentially zero in the mice themselves. There's very little background in these. That's a neutrophil. It doesn't project great. And then these two are lymphoblasts. So you have circulating CD4 positive lymphoblasts in these animals. These animals have undetectable lymph nodes in the control. Try as we might, we cannot pull out a lymph node from the NOG-SCIDs. The transduced, the adoptively transferred NOG-SCIDs have lymph nodes readily detectable with an infiltration of a fairly monomorphic population of CD4 positive lymphoid cells. And in parenchymal organs such as the liver, you have CD4 cells leaving vessels and entering parenchyma as well. So modeling a leukemia-like phenotype merely by one round of immune stimulation of these cells. I'm going to tell two other little stories here as we finish up. The long-term surviving cells continue to secrete fairly high levels of a number of cytokines. By about week 20, some of them are decreasing significantly. But again, even in the long-term setting, where the cells are, by DNA analysis for T-cell receptor, now oligoclonal. We can count with different T-cell receptor PCR reactions on the order of 30 or 40 different sorts of clones, those sorts of range. You get sustained production of cytokines associated, again, with essentially all of the different subtypes, again suggesting that Tax is also confusing the fate of these cells. I want to point out in particular IL-10, associated with T regulatory and suppressor function, and TGF-beta also associated, that's week 12 there, it got cut off, also associated with suppressor activity, Treg type function. And I'm highlighting those because of the question in the literature about what are the CD4 cells in HTLV-1 leukemia lymphoma. Or do they have a bias? Do they have a Th1, Th2 sort of bias to them in some way? And there's been some thought in the literature that they may actually have some T regulatory-like activity. So this is one paper from a Japanese group, but other individuals say that in ham-TSP you actually lose the Treg, and that's why you have more of an autoimmune type response. So Allison has looked in the long-term cells and identified increased levels of FOXP3, a transcription factor associated with Treg activity, and did a very preliminary experiment. I'm going to stress this. This was in outbred mice. It was in the transgenic strain. We're now repeating this in inbred strains that the tax has been moved on to, but asking whether the secretion of these Treg-type cytokines by the activated tax cells can affect the function of CD8 cells. Remember, CTL controls this infection very efficiently. In fact, in asymptomatic patients and patients with ham-TSP, up to 28% of circulating T cells are CTL against a single epitope of tax in vivo in people. So the question is, do tax-expressing cells defeat CD8 cells? Very simply. So the experiment was to ask if you immune-stimulated tax CD4 cells and mixed them with immune-activated CD8 cells, either tax or non-tax CD8 cells, will you suppress CD8 cell proliferation? So she's repeated these experiments, but again, they're in an outbred strain, and we're going to confirm this in an inbred background. But fundamentally, what you see here are activation markers. So activation of CD4 cells, showing that with immune stimulation, the tax and the control CD4 cells are both stimulated, and there are large numbers of stimulated cells. What you see then in the control mixture of CD4s and CD8s, you also get a substantial number of activated CD8s in this mixed culture of a control CD4 and CD8 cells. But when you mix tax CD4s with tax CD8s and we're going to have to, we've done it once in the outbred background, that's not meaningful. We're going to do it in the inbred background. So this is tax CD4 and tax CD8 T cells. You see a marked suppression of activation of CD8s. And this has been repeated in the outbred background, suggesting that not only does activation of tax expression induce proliferation, but it may actually inhibit CD8 response and CTL response. So this piece really summarized that immune activation of the LTR tax CD4 cells from this mouse model induces hyperproliferation and activation, associated with tax expression, and immortalization of these cells, potentially with a role of MCL1 in that immortalization. The shRNA experiments are about to be done, and that adoptive transfer causes a leukemia-like phenotype, that the cells themselves have a confused phenotype. And Allison has actually now seen single cells with transcription factors characteristic of both Th1 and Th2 cells, for example, and that the transformed cells may suppress CTL function. So the question we're now investigating in the last couple minutes, Céline Granier is working on, is why don't the mice themselves get lymphoma? And part of the answer to that is that we do see, and Céline has seen, I won't show you the data, that the tax does induce apoptosis in vivo in these animals. And actually, Bill Hall published something like that in a similar mouse in about 1998 or so. So there is some apoptosis. So you may have to get beyond tax-induced apoptosis in order to get oncogenesis. So what are possible collaborating factors enabling transformation? One would be T-cell activation itself, and we're investigating this with antigen-specific activation now in transgenic mice. A second would be secondary infections, and there's an extremely interesting clinical tidbit, which is that patients with Strongyloides stercolis, so a nematode infection of the GI tract, have both hyperstrongyloides syndrome as well as marked more rapid progression to any HTLV1 disease phenotype, be it leukemia or be it ham-TSP. So there may actually be a role of secondary infections. And finally, clearly, additional mutations, which we know happen in the host cells with all the alterations that are going on in those cells. And one of the approaches that we're taking is a screen. I don't have data on a gene per se, but I want to show you where we are because it's very encouraging. Celine has pursued the very commonly used retroviral insertional mutagenesis model, where she asks, Maloney-murine leukemia virus will induce CD4 cell lymphomas in mice, but it takes 3.5, 4, 5 months to get those lymphomas. What happens if she infects the tax mice? Will she accelerate induction of leukemogenesis, potentially through a collaboration of the low levels of tax expression that we have in the mice with the infecting virus and with genes that might be turned on by that virus through their integration site, through common integration sites? So very simply, newborn LTR tax or control littermates were infected with murine leukemia virus, IP, and Celine's been monitoring the mice, and here was our first mouse. So this is a mouse that was at about 2.5 months, well before the time period that, or even 2 months, well before the time period that Maloney itself had caused any disease, and that mouse had a 1.5 gram spleen as compared with a tax transgenic that was not infected. And this was a mesenteric lymph node looking almost like a piece of intestine, it was so large. In fact, here's the Kaplan-Meier plot. These are the Maloney mice now at 130 days with about 20% getting lymphomas, whereas by about 100 days, 50% of the tax mice had gotten lymphomas, and in fact, this was highly significant. So Celine is now in the course of trying to identify common insertion sites that might provide clues as to genes that can cooperate with tax to ultimately induce lymphomas in these mice. This is histology from I think that first mouse actually, control spleen with white pulp, red pulp, white pulp, red pulp, and instead these massive nodular lymphoma with infiltrating larger lymphocytes at higher power. There's some irregularity to the cells. There's a leukemia phenotype, and some of the cells have sort of lobulated nuclei, not perfect flower cells, but beginning to look somewhat like the flower cell phenotype. So that really is where I want to stop, but fundamentally that we're continuing efforts to try to induce disease in vivo, which obviously would really prove the model, but we think that there are real implications in this model that patients are infected initially and there's establishment of latency, strong cytotoxic T-cell response, but somehow induction of tax expression, potentially immune activation mediated, can lead you down a trail that ultimately will lead to this oligoclonal proliferation and ultimately to disease. So thank you very much, and I'm happy to take questions.
Questions?
It's late.
So do you think that in the human disease, it is the activation of T cells later on in life, or the auto-activation, so that people, for instance, I mean, you have a chance to test the antigens that they go against and see if there's auto-activation?
Yeah, I think that's, that's a really interesting question. The T-cell receptor itself is down-modulated on most of the tumors. What the antigen specificity is of the more autoimmune-like diseases isn't known, whether it's truly sort of a cross-reactivity with self-antigen or whether it's a bystander effect like HIV. That's not known. Again, there was, for sort of students who are here, I always quote this, David Baltimore at an anniversary of the discovery of reverse transcriptase made the point to the students in the audience that science proceeds by analogy. And a lot of this is conditioned by analogies to things that have happened in other viruses. So I think it's really increasingly clear in HIV that immune activation really does play a role in sort of activating those latent reservoirs, and that would be the argument that something like this does happen. But it could be of real interest to look, particularly in the autoimmune diseases, about what the reactivity of those cells is.
The other thing that is interesting is as you move in vitro, and now you see something you don't see in vivo, it'd be interesting to add back Tregs, or add back, I mean, it's a little bit like EBV in the sense that if you have T cells around, you don't get the immortalized cell to grow out as well as if you don't have T cells around.
Right,
so
absolutely.
You could reconstitute in an interesting way.
Yeah, I think doing this now, so the experiment that Celine is doing now is doing in vivo immune activation. We put the LTR tax onto a mouse where all the T cell receptors in the mouse are against ovalbumin peptide, and it's CD4 specific. So what we're going to do now is stimulate with ovalbumin and see if we can really sort of drive in a system where there will be potentially all kinds of T helper cells around of all the different flavors. The issue of what's going on in vivo with these different flavors of T cells has been so confusing that we think the mouse system may actually help us sort that out. If we get some idea, we can then go back to patients because we think it may be confusing because the cells themselves are expressing multiple phenotypes. They're not expressing sort of the canonical Th1, Th2 sort of phenotype.
You have a question here.
Again, in reference to the EBV and the EBNA story, do you have any data whether the tax is required for the persistence of malignancy in your system?
So this one's very clear. It's not our data, it's others. Tax is not required for persistence of malignancy. In fact, expression of tax is probably selected against during this process. Most ATL cells, almost none express tax, and many have actual deletions in the genome or mutations that block tax expression. So this is an early process. There's no question about it. Again, that sort of potent CTL might really select against continued tax expression. So there is a fundamental difference. Well, I don't know how much of a difference. Some things are probably similar and some are probably different in that sense. You probably don't express a lot of the sort of more surface antigens in the EBV setting either.
Is the shift from oligoclonality to monoclonality in immune selection, or is it inherent in the cells?
Yeah, I don't think the answer to that is really known. The other thing that's really unclear in vivo, although it's very easy to talk about, is that you go from IL-2 dependence to IL-2 independence. That appears to be true, but again, there's some data suggesting that even tumor cells may have IL-2 dependence in culture as well. So those are really good points. It's surprising some of the holes that are really there in the understanding of the in vivo disease.
In a somewhat different vein, I know in Japan, public health has been really successful in limiting the spread of the virus by urging positive women not to breastfeed.
Correct.
Does that work elsewhere in the world?
I think it would. Yeah, I mean, I think it's. I don't know what's. I'm not sure enough. I'm not as aware of the public health sort of infrastructure. I know in Jamaica there's a stronger public health infrastructure, and there's been a push towards that. I don't know how successful it's been. In the HIV setting, there's been this, this sort of difficulty because not breastfeeding also predisposes to a whole array of other diseases, and in fact, there have been kids who've died of contamination of formula, et cetera. So it's a lot harder to implement in some of the more third world, countries in this sense. But it's been you're absolutely right, very successful in Japan. Brazil is a budding epicenter of this. I mean, there's something like 0.1% of all blood donors in Rio are positive or something like that, and Rio's a big city. So there's a lot of disease in Brazil, and that'll be a place where these sorts of things really need to be implemented.
I think someone once said this is a virus that was ready to die out until blood transfusions brought it back. Is that right?
I think if you really couple breast milk, with, well, there is sexual transmission though. Yeah. And in fact, but again, it's not a very hardy virus. In fact, most of the transmission is male to female. I mean, and it's transmission of lymphocytes that are infected again, because the virus is such a weak virus. I mean, the silver lining in this is thank God it's such a weak virus compared with HIV, but there still are, 15 to 25 million people infected. but it is, you can't really grow it as cell-free virus. It's very difficult. And the CDC folks would sort of joke that it's hardly a virus. It's very hard to do some of these properties. I don't know if you'd argue that it was going to die out. Allison shows the picture. There's wonderful anthropology of this virus. The virus has been found in Andean mummies. So, multiple times. So it clearly came down across the Bering Strait as well. There are parenthetically, and this bears on some of the things I just watching CNN and the infections and epidemic era that we're in now. There's an HTLV3 and 4 that are related to HTLV1, 2. This is not old Gallows HTLV3. These are viruses related, which are bushmeat transmissions. They are monkey viruses that have transferred into single people, small numbers of people, probably through, cuts and butchering, bushmeat, et cetera. But as yet, no real evidence of person-to-person spread. But there's a cauldron out there of different viruses waiting to cross species lines. Yeah.
Well, on that happy note,
We didn't talk about swine flu.
that's right. Well, I want to thank all the speakers and the audience as well. Thank you.